Hello, everyone. Welcome to today's live broadcast, Detection and Analysis of Circulating Tumor Cells, Challenges and Solutions. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. Whether your passion for answers takes you to a laboratory bench or a hospital bedside, the pursuit often begins with a blood or tissue sample. The deepest mysteries of disease and other biological processes, the answers you seek are encoded in the building blocks of life, DNA. Yet the work of learning what these molecules can tell you is quite a journey, and we at QIAGEN understand the challenges associated with this quest. Our commitment is to enable you to quickly and reliably reach your goal of useful, actionable insights. To this end, QIAGEN offers you innovative solutions that cover every single step along this journey, not just technologies, but bridges from samples to insights. Samples to insights means QIAGEN offers you the industry's most reliable sample technologies because samples matter to your success. Our top quality assays and panels enable you to accurately analyze and identify diseases and genetic variations. Our bioinformatics software and curated knowledge bases transform your raw data into relevant, actionable findings. And our automation solutions provide you seamless and cost-effective workflows. Sample to insights. The insights you gain may lead to one small step or a giant leap forward for science and healthcare. Partnering with QIAGEN, your work can make a difference. For more information, please visit QIAGEN.com. We have a few important announcements before we begin. The webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Additionally, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left corner. This is an educational webinar, and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process for obtaining your credits. I would now like to introduce today's speaker, Dr. Siegfried Hauch, Director of Liquid Biopsy and Site Manager, QIAGEN, Hanover, Germany. Dr. Hauch studied biology at the Albert Einstein University in Ulm and received his PhD in 1997 in plant physiology and molecular biology at the Eberhard Karls University, Tübingen. Until 2001, he was project manager for imaging systems and microarray technologies at Biozyme Diagnostics GmbH, and changed thereafter to AdnaGen as marketing manager in 2001. In addition to his marketing responsibilities, he was Director of Research and Development since 2006 to build and to secure and enhance AdnaGen's circulating tumor cell business. And after the acquisition by Alere Incorporated in 2010, he was holding the position of CSO and General Manager at Alere AdnaGen GmbH. Since 2015, Dr. Hauch has served as Director of Liquid Biopsy and Site Manager at QIAGEN Hanover GmbH. I will now turn it over to Dr. Hauch for his presentation. Doctor?
Yeah, thank you very much for this kind introduction. And thank you also for everybody in the auditory for taking the time to listen what I have to tell you about circulating tumor cells, a member of the liquid biopsy family and its molecular characterization. And as you heard, I'm working since nearly 16 years now in that field, and I would like to share with you what I observed on the way in terms of challenges and how we found solutions to better be able to get meaningful information out of circulating tumor cells in patients' blood. First of all, a little disclaimer to make sure that you understand that what we are talking about today is really research. There is no intention to make you think about using this information for real patient diagnostics. Thank you very much for that. Okay, let's start with the AdnaTest as such. So this is the test I was able to develop with my team, and in order to understand the challenges and solutions we found on the way, you probably need to understand a little bit more about the test principle as such. Okay, this is now, okay. So what is this test doing actually? So how do we address and access the circulating tumor cells? It is actually starting with something you might already know. It's immunomagnetic capturing in a first instance that we use to enrich the cells from the blood of a patient. Enrichment means, by the way, here that it is not pure CTCs you get out. We will come later to that because it's one of the challenges that we always, whatever method you are looking there, you have a contamination of a certain amount of leukocytes which needs to be addressed and needs to be taken into consideration if we think about molecular characterization of such cells. What is the difference here that makes us different from other immunomagnetic technologies? And this is the CoCP principle. The CoCP principle means combination of combinations principle. What is that? One of the secrets here is that the first combination of these combinations is a combination of antibodies directed against cell surface antigens on tumor cells. And when I say a mixture, I mean that we are not only using AirCam, as many others do, but we use several antibodies, up to 4, to capture the cells. The reason why we use this mixture is that already in 2004 we realized, and we called it at that time, tumor cell variability, which is today known as epithelial to mesenchymal transition, which means in fact that the tumor cells, once they enter the circulation, may lose certain specific antigens they had before and change their phenotype into a mesenchymal-like. And for instance, in many cases, 30% of the cases in breast cancer, for example, AdCam gets lost and replaced by other more mesenchymal-like antigens. And using this mixture, we make sure that we will not lose the cells if this is happening. This is then followed not by visualization of the cells, but direct lysis of the cells, mRNA isolation, reverse transcription, and PCR. And again, and this is the second combination, we are not only looking for one tumor-associated RNA species, we are looking for several marker genes if they are expressed or not. And the rule is, if one of these genes is overexpressed, this can be taken as a proof, first of all, that the cell or the signal we got comes from a circulating tumor cell, and we get more information even out of that, but this I will show you later. The AdnaTest, as it is today, is quite simple benchtop assay. It's a manual assay consisting of several incubation steps. It starts with blood sampling. The blood is incubated then with the immunomagnetic bead mixture for 30 minutes, and this is followed then by several washing steps to get rid of most of the leukocytes and all the red blood cells. To give you here already a kind of an idea of the purity we get out here. So after the whole procedure, the amount of contaminating leukocytes ranges in the range about 1,000 leukocytes. By using special procedure, which I will mention later, we can even reduce that to 150 leukocytes, and this already gives you access to the overexpression profile of many genes that are quite special for CTCs and not for the contaminating leukocytes. And all that is then followed by kind of a molecular PCR analysis. It could be an adjuvant, for example, you use for that, and you make kind of a capillary gel electrophoresis, or what we do now, of course, as you can imagine, we do qPCR to get a better quantification of the fragment concentrations and the overexpression profile. Okay, that is the tests available so far, and as you can see, we developed tests for breast, prostate, colorectal, and ovarian cancer. But also, since we knew about this EMT and stem cell issue, this phenotype change of the cells, we especially also developed kit systems to address especially these kind of cells, and we got very interesting results out of that. And this is the AdnaTest EMT1 and EMT2 and stem cell. And you see the target genes, which means the RNA genes that we are analyzing here on the right hand in this table here. Okay, let's come to the first challenge. And I already briefly mentioned that. This is the AdCam problem. 16 years ago, actually in 2004, the world thought that AdCam is the best marker to enrich and capture circulating tumor cells, which is partly true. But as I told you, this is unfortunately not true all the time, and this is also different in many cancers. And by using this immunomagnetic antibody bead mixture, you really can kind of overcome, at least most, you can overcome most of this challenge here. I would not say the mixture will probably not miss also here and there a cell that would even not have one of the 3 genes or so that we are using here. But I would say in most of the cases, and these our results show quite clearly, at least the increase in sensitivity is obvious. And see here the first example where we did a comparison in colorectal cancer, comparing an only AdCam using immunomagnetic method with the CoCP principal method of the AdnaTest. And you see there that the number of samples positive with the only AdCam was in that case 20%, and the corresponding samples showed an 80% positivity for circulating tumor cells using the AdnaTest colorectal cancer. This might be due to the fact that we only in this test, the antibody mixture consists only of different AdCam variants, different AdCam antibodies, and already that increases the sensitivity, but also it might be the second combination, the fact that we are using 3 antigens, 3 genes to identify positivity, which is AdCam overexpression, EGFR overexpression, and CEA overexpression. And also that combination contributes, as you can imagine, to the increasing sensitivity. The same we can see even in prostate cancer, and this was quite surprising to me because I thought prostate cancer is one of the cancers that is most likely overall AdCam positive. And interestingly, it was not the case. So in this little study we did at the Memorial Sloan Kettering in New York, we found when we compared again our prostate cancer capturing using anti-HER2 and anti-AdCam in combination, it was very similar in cases where the cell search system using only AdCam identified cells, but we found a significant amount of positivity in circulating tumor cells, also in the fraction where cell search didn't find anything. So that tells me also in prostate cancer as a slow-growing cancer, there is a certain amount of cells that might undergo EMT and stemness, and this can be identified by using the AdnaTest breast cancer in quite a good manner. In breast cancer, we also can see that this was analyzed by Andreopoulou et al. At the MD Anderson quite a while ago. and published in 2012. You can also see the benefit of the use of a combination of different MUC1 variants and AdCam antibodies, and the increase against AdCam only is from 36 to 53%, giving you an idea about how many EMT-like cells in breast cancer might be lost if you only use the AdCam. And this increase might also be due again to the second combination, which is AdCam MUC1 and HER2 on the RNA level, since if one of these is positive and not only AdCam alone, you have a positive finding of the CTCs, positive result. And super interesting, this is a very recent result published this year in Nature Scientific Reports in lung cancer, when we compared the AdnaTest EMT2, actually, which was not really dedicated to lung cancer, but using antibodies against AdCam, HER2, and EGFR in combination, and we compared that again to a pure AdCam-based isolation method. You can see that we get a 100% better sensitivity, an increase from 15 to 30% over all the stages of samples that we had here. Only if we analyze PI3K, AKT2, and ALDH1 and use the antibody mixture for capturing. Interestingly, by the way, this is not really clear here in that picture, therefore I tell you, you see that 80% of the cells we are captured here were ALDH1 positive, giving you a very good idea about that. Actually, most of the CTCs that we captured by using the antibody mix was already probably in a tumor stem cell-like state, or at least in the EMT state. Okay. Now we have solved these sensitivity issues due to the fact that we in the past underestimated the variability of the CTCs and the tendency of these cells to mimic themselves or mimic their epithelial origin by undergoing EMT. And the second bias, if we now think about molecular characterization, is the fact that's most best described probably with purity. I do not know so many technologies, actually I do not know any technology that is really able to purify CTCs, really. There is always contaminating leukocytes still present. And if you think about it, we have in a 5 milliliter blood sample, you have 5 times 10 to the 7th leukocytes there, and you can reduce it already to 1,000. This is a dramatic increase in purity. Nevertheless, certain genes that we would like to analyze on the transcriptome level will also be present in leukocytes. And the more leukocytes we have, the more bias we may get, at least in certain genes. How could we circumvent that probably? This is only to illustrate a little bit. This is a leukocyte spiking experiment that I can show you here, where you really can see for a certain number of genes that might be of interest in CTC research, like AKT2, Notch, PAR, PI3K as EMT markers, but also interesting genes like the HER2 and the SRC1 that has probably one day a kind of a therapy-related meaning. You can see that the leukocytes indeed show an expression profile for these genes. And this must be taken into consideration, really, if we would like to exclude such a bias from our CTC results. First of all, for the normal AdnaTest combinations, you can see that to reduce the leukocyte count below 1,000 leukocytes, if you analyze genes like AdCam, HER2, or MUC1, is good enough. So 1,000 leukocytes allow you to address these genes and allow you to analyze these profiles even in a follow-up during a trial or something it looks quite stable. You see the specificity here is coming close to 100%, whereas the recovery ranges for 2 cells at 95% or 90%, and for 5 cells is coming close to 100%. So for the selected genes that we are using in the traditional kits, the leukocyte problem is already not biasing out our results. But what is about more? We do not only want to know really is the tumor cell there or not. We would like to analyze more genes, like giving pathway information, which pathway, molecular pathway switched on and off, what would be potential therapeutic targets of the future, what is the tumor biology. I would like to have as much genes as possible, and in that case 1,000 leukocytes turned out to be really too much. It took quite a while until we found a solution how we can reduce the leukocytes even by another log to 100 to 150 leukocytes. And this is possible due to a special washing buffer we developed, and that which we use instead of PBS during the first washing steps on the magnetic beads. Illustrated here, you can see the CD45 signal that we get with AdnaWash and without AdnaWash, and you can clearly see that the fragment concentration without the AdnaWash is dramatically higher, given here in nanogram per microliter fragment concentration if you don't use the AdnaWash, and it gets dramatically reduced if you use the AdnaWash buffer during the cell enrichment process. And doing so, doing that, we were able to address, at least in the first instance, the EMT-related genes. Let me give you a short overview what I mean when I'm talking about EMT and tumor stemness. The theory behind that is that in a tumor, the priority of the cells you can get there are kind of differentiated cells and are kind of actually not actively dividing cells anymore. Probably they are already necrotic. The driver of the tumor and the driver of metastasization is not this kind of cells. The driver of metastasization is a very small population of cells in the tumor tissue known today as tumor stem cells. And these tumor stem cells, for whatever reason, one day get out of the tumor, walk into the or enter the circulation actually as a mesenchymal cell, and in the circulation these cells transform to tumor stem cells with the capacity to form metastases. How can we address such cells, or how can we say that? The first finding we had in that view is that actually we found even in 2006 already that there is a difference actually if we analyze tumor tissue and the CTCs, for example, viewing to HER2. If we stain the tissue for HER2 and compare it to the corresponding CTCs and their HER2 overexpression profile, we find 50% of the cells or 50% of the samples are comparable, and another 50% are different. So you have roughly 30% of the CTCs in that study was negative for HER2, whereas the histology in the tissue was positive. But more interestingly, you had 21% HER2 positive CTCs, whereas the histology of the tissue was negative. That means, in principle, that there is a number of cells that redevelops or expresses HER2 as a growth factor, probably as a kind of a resistance metabolism that was not there before but is induced by a therapeutic intervention. And also genes like the estrogen receptor and the progesterone receptor, which can be addressed using the AdnaWash suddenly, show kind of variations in tissue versus the circulating tumor cells. But other than HER2, where we see a high proportion of de novo developed expression which was not there in the tissue, the estrogen receptor and the progesterone receptor get strongly downregulated once the cells go into the circulation, which is again a kind of a marker what tumor stem cell means, because tumor stem cell is not a highly proliferative cell. A tumor stem cell is a cell that has increased survival mechanism and has a metastatic capacity, but in the bloodstream it's getting silenced, and this might be kind of described here by the downregulation of the estrogen receptor and the progesterone receptor. And if we then look for EMT and tumor stemness related markers really, we see indeed we have been right. And these are results with AdnaWash prepared cells, because without that it's not possible to address these genes like Twist, AKT2 and PI3K, which are known EMT markers in breast cancer, or the ALDH1, which is a stem cell marker in breast cancer and many others. And what we see here, what really happens is that if CTCs are in the circulation, I would say based on these results here, nearly all of them, at least 80% of them, taking everything together, are either EMT-like or tumor stem cell-like. That means, in other words, that metastasization is that a number of these low-frequency cells that have already the stemness capability in the tissue is really the phenotype of cells that leaves the tumor, enters the circulation, and is the seat of metastasization, at least based on these results here. What else can we do, if we see such results? And this is a very simple thing. Once you have such results and you know now you have the methodology and could overcome the AirCam problem and the leukocyte bias, and you have now suddenly access to probably a great number of genes in parallel, only a few genes is not enough. And this was the reason actually why we decided to go for a panel development. We wanted to know more. And the first panel that we developed here, which is, by the way, not commercially available, this was really the first try, but already successful, was a panel consisting of epithelial-like genes, genes that are in EMT metabolism important, and genes of the tumor stemness family like CD44 or ALDH1. And we clearly could see that using the AdnaTest enrichment process and the cDNA we developed with these kids was enough material to really address up to, what is it here? Let me say 20 genes or so, because this is the random number that we usually use if we do now ADNA panels, or if we develop now ADNA panels. And you can see in this little preliminary trial in 18 patients, we got a real nice differential expression of these different genes and could address that very nicely. And also in the individual situation, you can see the differential expression of the genes gives you clear information, or at least you could interpret clearly information out of that. And this is shown here in a kind of casuistics in 4 samples. So we have patient sample G, where we can see in this panel only AirCam is overexpressed in the cDNA we got from that patient. And in patient P, we have only HER2. The AdCam is not there, but also not other genes of the EMT pathways or the tumor stemness pathways. Whereas patient M is, in my eyes, a clear tumor stem cell-like phenotype that we captured here. CD44 is there, Twist is there, mTOR, PI3K, all indications that both EMT metabolism, but also tumor stem cell-like metabolism is switched on. And another interesting profile is patient E, because in the first instance you would say, yeah, CD44 is on, therefore this is a tumor stem cell-like cell, but we also see AdCam suddenly. We see P10, which is actually an inhibitor, an intrinsic inhibitor of the EMT pathway, of the PI3K pathway. And we see suddenly KRAS that could give us an idea about if it's not the PI3K pathway in breast cancer that is making the metabolism of this cell dangerous, is KRAS probably a resistance mechanism? Can we see that applying these kind of panels? We hope yes. At least it's an invitation to do more research in that direction, to see what is really happening in this cell during a therapy, for example, or as a consequence of therapy resistance development, or as a consequence of therapy response even. How do the cells react on that? Is there a metabolic change, and a change into what direction, and would this give us in future necessary information to be able to better react on that, to earlier treat patients with the right targets and the right drugs developed for the certain targets? Our wish was, when we did this research, we wanted to be able at least to kind of build a scale where we could sort patients in that might have CTCs that are not really dangerous, that are probably not seeds of metastasization, which is the more epithelial likes, and we wanted to find out another group of patients that is develop a more aggressive cell type, and where on the scale could we build in the patients here. And in our case, it's very clear patient G with the only AdCam was an example for the more epithelial-like. Patient P, you remember that was the one where we had the only HER2 expression. I would see not already EMT-like, but on the way there probably. Patient E is the more EMT-like, and patient M the more stem cell-like. And the big question that comes here now, the position on that scale, is that indicative about prognostic risk of the patient or not? And this is a question of studies that have to be done really to confirm it. Other panels we developed afterwards based on these results, because we also had genes on the first panel that never appeared in breast cancer. It was the first panel that we developed here for breast cancer that will be probably commercially available soon. And this is consisting of AdCam, ERCC1, HER2, AKT, EGFR, HER3, PI3K, ADH1, and Aurora kinase. And when we did first studies with that panel, we first wanted to know, is there a change in the profile of these genes if we look at it before and after therapy? And we found, yeah, there were slight changes in terms of EGFR and HER2. So HER2 is upregulated after therapy. EGFR in these cases was downregulated after therapy in frequency. ADH1 as expected. So if there were cells after therapy still there, a high proportion of them was ADH1 positive. And the interesting thing here is Aurora kinase was exclusively overexpressed only in cells we found after therapy, but never before. The more important question for us was there a correlation with therapy response or not? And here the picture was a little different. We grouped the genes we had there in epithelial-related genes, in receptor expression genes, in resistance markers, in EMT-likes, and in stem cell-likes. And indeed, also the epithelial-like plays a role, as it looks, in the group of the non-responders, as you can see here. Whereas the receptor expression did not differ, but the resistance markers like ERCC1 and Aurora kinase were correlated with the non-response group, whereas the responders were lower in frequency. And surprise, surprise, also the stem cell likes again follow this route. nearly exclusively observed in the non-responder groups, whereas nearly not expressed in the responder groups. That was not only nice results, but also statistically, in many cases, significant. You can see here that after therapy, epithelial likes were significantly present in after therapy in these cases. The stem cell likes also after therapy, but we found also combinations like EGFR and aurora kinase that was already kind of indicative before therapy, and this is a very interesting finding because the question is appearing here, would we really like to only have a monitoring, or would we really like to only have a kind of a prognostic information, or would CTC probably also be able to give us a kind of predictive information before a therapy started, which might be the right regimen to give or not. And this is the first indications here. If you look at combinations like EGFR or AdCam in combination with aurora kinase, or all the three, all they were significant correlated before therapy, which gives us at least the idea that it might be possible that the molecular characterization of CTCs might give us, instead of prognostic information, also predictive information in the future. One could do it even one step further. You have seen that we developed a panel with 20 genes. Others developed panels with even more genes. And this is an example coming from Katarina Kolostova at the Eberhard Karls University in Prague. They had a panel there for, there is a typo I see, it was actually 48 genes they analyzed for, and they made the following study. First of all, they generated cDNA by using the AdnaTests EMT1 and stem cell, and generated the cDNA and analyzed using the Agilent for positivity or not, only that. In parallel, they also analyzed the cDNA for their 48 gene expression panel. Also, they took FFPE tissue and analyzed for the same panel and compared it to the CTC findings. And this is what they found. And this is a very interesting result, if you ask me. So that's probably give us an idea, at least a vision, what is really happening during a metastasization process, and how can we make research with that. You can see here in light blue the partial clustering results of the tissue. And in black, you can see the positive CTC findings that we had in metastatic disease. And in green, you see the positive CTC findings in primary disease. And in what is that purple, you see the negative, AdnaTest negative samples. First of all, that tells us, and that's nice, the negative samples are grouping away from all the other groups. That gives you an idea about the specificity of the tests. If we don't find the CTCs, they do not group to the CTC group, and also not to the tissue. And also the metastatic CTCs have only a minimal overlap with the tissue. That was surprising, actually, for me, but kind of following the route that really during the metastasization process significant changes apply here, and probably this is driven by the phenotype of the CTCs that go in the circulation and form the metastases. And the CTCs in the primary situation, they overlap both groups, the tissue and the metastatic CTCs. And here comes now my idea. I'm not sure if it's true, but it's worth thinking about it. Do we see here, if we look in the primary CTC group, and if we take the subgroup of primary CTCs that fall into the FFPE tissue sample group, is that a CTC type that is still expressing kind of a a non-dangerous tissue-like phenotype, but already you find CTCs in the primary group that have a profile that overlaps with the metastatic disease. Is that the subgroup in the primary that gives you already the information that these CTCs are on the way or already kind of acquired metastatic metabolism ready to form metastases? Interesting findings here. We keep on researching on that. Let's see, in a year from now, we will have more information probably. Let me summarize. The molecular detection of circulating tumor cells have proven to be a highly sensitive method and very specific by using the combination of combinations principle, making sure that we get the maximum sensitivity out without losing the specificity as I was able to show you in the case of AdnaTest breast. The CoCP principle has turned out to really improve the sensitivity substantially, making again as a proof that it's really true that certain genes get lost once the cells get into the circulation, especially if some phenomenon like EMT or tumor stemness appears. The molecular characterization of CTCs has been proven to be able to identify EMT and tumor stem cell related phenotypes by means of molecular biology and gives an indication about molecular pathways that might be involved in metastasis formation, but more importantly, that might be involved in resistance formation, because that could in future give us ideas about how to overcome resistance really. If we see what is the reason for the resistance, we have at least an idea about what could we do to overcome the resistance and to help the patient. And the most important final information I could give you today on this call, the AdnaTest is an open platform that enables you, the researchers, to have your own ideas. What would you like to analyze? What kind of tumor type would you like to analyze? In what direction? Would it be more kind of targeted analysis in terms of drug resistance or something? Would it be into the direction of better characterization of the overall CTC metabolism? Everything is open. You have the cDNA. You can develop your own tests and you can find your own results. And this is the most important message which I would like to place here as a final. And with that, I thank you for your attendance. I thank you for your patience following me all the time, and I'm ready now for questions if there are some.
Thank you, Dr. Hauch, for that informative presentation. And before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, can the AdnaTest be used as a mouse lymphoma model, or would you need to optimize antibody as the MSC marker as they might be different?
Yeah, that's the case for sure. So if we talk about lymphoma, this has nothing to do with epithelial cancers, which I was demonstrating you today. Nevertheless, I mean, the principle stays in fact the same. So one needs to find the relevant markers that are useful for capturing, and of course relevant molecular markers we would like to see then afterwards in their expression profile. In principle, it's possible. In reality, we didn't focus on lymphoma and such kind of cancers because we were more focusing on the epithelial ones and the solid ones.
Excellent, thank you. Our second question asks, can you discriminate between metastatic and non-metastatic patients?
Okay. If I understood you right, it means metastatic or non-metastatic patients?
Yes.
Okay. Yes. A simple answer, yes. So there is a nice piece of work we did recently in the group of the University of Essen, where we could show that the profiles that we find in the non-metastatic disease, if there are CTCs, has indeed big differences against the metastatic profile. And even more, what we found there seems also to be in the metastatic situation a kind of preference genes that are related to the site of metastasization. That means also you can use the metastatic profile to identify, statistically I have to say, it's not an identification for sure, but there's a statistical correlation to the site of metastasization even.
Thank you. We have an additional question. Can the AdnaTest system detect 1 CTC in 1 ml blood?
Yes. That's a simple question and a simple answer.
Perfect. How sensitive is the AdnaTest?
Yeah, sensitivity is how should we describe sensitivity? We can describe it in 2 ways. One way is that we would have a technical sensitivity for which the name is recovery. If I do spiking experiments using cultured cells, I get recovery rate of 90% if I spike 2 cells in 5 ml of blood. So that is the technical sensitivity. If we now go to the clinical sensitivity, so that means what is the frequency, the rate of the cells we get if we, for example, look into a cohort of metastatic patients or primary patients. The clinical sensitivity in the primary situation over all the cancers I demonstrated here, might it be colon, ovarian, breast, or prostate or such, is between 20 and 30% in the primary situation. Means 30% of the patients before surgery or something are positive for CTCs usually. In the metastatic situation, this can go up to 70% clinical sensitivity, again dependent on the situation. So when I say 70-75%, that means not treatment naive, but also in the between the treatments or so, at least 4 weeks no treatment, then it can go up to 75% positivity for CTCs in metastatic samples.
Okay, thank you for that. Our next question, how early or at which stage in cancer development is it possible to detect CTCs in patients' blood?
That's a good question. Thank you for that. I had it only once in the last 4 years, but it's a really good question. So if your tumor is small or large, if there is already nodal involvement or not, CTCs, the frequency of CTCs is not correlated to that. You find the same frequency in, let's say, T1, N0, M0 breast cancer patients, as well as in T3, N2, and so. That means circulating tumor cells are an independent marker over the normal staging parameters.
Okay, thank you. Next question. In addition to CTC count and disease, how can you perform tumor-specific mutation measurements in CTCs? Yeah, so of course we did research on that, and we were able actually to do it in the case of colorectal cancer. We were able to really find CTCs with KRAS mutations. In that case, it was codon 12. That had been actually very preliminary experiments, I have to admit. So that was not our focus in the past, but we do more and more work on that because we have seen at least the proof of principle for colorectal cancer was there. And at least also we got an interesting finding here in that case because we could see that the KRAS mutations we found in the CTCs were not necessarily also found in the tissue, and vice versa. So we found in this little study published by Paola Gazzaniga
or Raimondi et al. From the Policlinico in Rome, they found tissue that was positive for KRAS codon 12 mutations, but the CTCs were not, but also the other way around. We found tissue initially negative for the mutation, but suddenly KRAS mutations were there in the circulating tumor cells, which might be the case because of the, let's say, these tumor stem cells are so rare in tissue that you might overlook them. But once you capture them in the circulation, specifically by using the AdnaTests, for example, there might be a higher chance to find the mutated ones really in the circulation.
Thank you. Excellent. If there are no more questions, I would like once again to thank you, Dr. Hauch, for his presentation. Dr. Hauch, do you have any final closing comments?
Yes, I probably have two comments. So first of all, again, thank you for giving me the chance to have this webinar today. Second, the auditory, thanks again for your patience and for your contribution in terms of questions. And the third, hey, let's do research together. It's a good field, and we are getting further and further. And for the sake of the patient, we should find good solutions in the future.
Thank you so much. I would also like to thank our sponsor, QIAGEN, for making today's educational webcast possible. Today's webcast will be available on demand viewing through February 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you so much, and see you next time from LabRoots. Goodbye.
Bye-bye.